So Jessica is a postdoctoral researcher at the South African Institute for Aquatic Biodiversity, who uses genomic tools to study the evolution and ecology of jacks and trevallies to inform international fisheries management. She received her PhD from Yale University in 2019, where she was conducting extensive research on trevallies in Seychelles and other Western Indian Ocean nations, thanks to the help of several citizens. So her presentation will be about the genetic connectivity and spatial trophic variability of giant trevally and bluefin trevally across Seychelles. So Jessica, over to you.
Hi,
Okay.
everyone. Thanks so much for that kind introduction, and to all the organizers, especially Sheena, Tama, and Sophie. I really enjoyed the talks over the last 2 days. And today I'm going to talk about 2 projects on 2 iconic trevally fish species, which has comprised my dissertation work over the last 5 years. So as many of you probably know, in Seychelles, the trevallies and the kingfish are significant for the local fishery catches. If you walk into the Victoria market, you're going to see bludger trevally and yellow spotted and big eye, several species that are important for food fish at a local level. But there's other species that are very important to Seychelles for tourism, specifically the giant and the bluefin trevallies. These are 2 of the most highly targeted recreational fishery species in the outer islands and to some extent in the inner islands. And for example, on Farquhar Atoll, 100% of fly fishing guides rank trevallies as #1, their most highly targeted species. And we know that from research done by Sheena Tama as part of her master's thesis, where she conducted a socioeconomic survey of the fly fishing industry. And this kind of work has allowed us to kind of better understand what species are important for these fishery stakeholders, like the kingfishes, and what kind of data still needs to be collected. One of the biggest challenges is that a lot of kind of fundamental biological information is still unanswered for the trevallies, both in Seychelles and actually throughout a lot of their ranges. So the giant and bluefin trevallies are found throughout the tropical and subtropical Indian and Pacific Oceans. And as you can see on this map, they overlap a lot throughout most of their ranges, which is the overlap is shown in purple. And they are coastal fishes that are associated with coral and rocky reefs. And so given some recent work in the Western Indian Ocean and previous work like in Hawaii on trevally movement behavior, one of the big questions that remained unaddressed was the genetic connectivity. So when I started working on these, the only place where anyone had studied the genetic connectivity was in Hawaii, which is a very small part of their range. And so I started out wanting to address connectivity in Seychelles and the Western Indian Ocean, but then this has quickly grown into a global project. So the questions that we've been looking at include, what's the extent of connectivity across their range? Is there geographic structure? Are these species impacted by the typical biogeographic barriers that affect other coastal and reef-associated fishes? But they're quite large fish, and found throughout many locations. And so collecting, a sufficient number of samples has been challenging, partially because they're not, heavily targeted by industrial fisheries. But luckily, because they're highly prized by sport fisheries, we've been able to collaborate with sport fishermen and citizen scientists across 14 different countries in the Indo-Pacific. Really fruitful and in collaboration where readers are sampling and they get into it on Insta client samples, and science has been engaging of this in critical in Seychelles, especially in the Seychelles fishing and SFA have been really helpful as private charter operators. We collected hundreds of samples in different localities across black being giant and the blue numbers indicating bluefin. These are just actually a small subset of all the samples. We even have several hundred samples from Seychelles alone, but I needed to sort of take a subset to compare for the genetic with other locations that have lower sampling numbers. I'm not going to go into too much detail in the methods, but we use SNP data. And what's really exciting is that some collaborators have actually sequenced the entire genomes of both of these species, which opens up a whole new slew of potential research looking at adaptation and selection and really delving into these. I'm just going to jump right into the results. These are PCA plots that are showing both species. And each individual point is one fish. And so most clustered together, those individuals are more genetically similar to one another. And so these kind of ellipses don't present anything like significance. I just drew them, highlight that there's multiple populations of both species. So in the case of the bluefin trevally, it's 3 putative populations, one in Hawaii, Pacific, and then the inclusive, I'm going to point out, and then for you also see a separate population in Hawaii and the island of Kiribati, Western Asian population, and there's this sort of gradient in the Western, and I'll show that in detail. Here's the bluefin trevally, and on the top is a figure that we call structure plots. You may have seen these before. And basically each bar represents one individual fish, and the colors represent the proportions of an individual's genome that originate from some number of ancestral gene pools. So these are ordered from west to east, from South Africa to Hawaii. And you can see that there's this evidence here of this separate putative population in Hawaii. There's some kind of drifting that I won't talk about now, but you can ask me about later. And then this other sort of indication of genetic similarity in the Indian Ocean as well, from South Africa to Maldives. But everything in the Western Indian Ocean is pretty panmictic. Same for the giant trevally. You see this evidence of a strong kind of genetically distinct Central Pacific population. And then it's actually more easily visible if you look at pairwise measures of genetic differentiation. These are called FST values. And basically, this plot on the bottom left shows that the Hawaii and Kiribati are more genetically distinct from everywhere else. And then you see this sort of gradient as you're moving westward, with the Western Indian Ocean being very genetically similar. So individuals from these sites are very genetically similar to one another. So these results, first of all, point to possible differences in life history characteristics between these two species, maybe in pelagic larval duration, juvenile survival, or spawning time. They're slightly different patterns, but I think more interestingly, it shows that these two species more closely resemble patterns of open ocean pelagic species like sharks and tunas that are either highly connected or show substructure at the level of ocean basins rather than most reef-associated fishes. And this is interesting because we know giant trevallies in particular are highly territorial and pretty resident as adults. So this leads us to then the question of, if they're genetically similar in the Indian Ocean, are they ecologically similar? And do they play the same functional role across different habitat types? And so the following slides, I'm just going to be including giant trevally because we didn't have enough bluefin trevally samples to do as large of a comparative analysis. So one way you can look at this, of course, at diet and ecological function is through sediment content analysis. That's tricky when you're working with sport fishermen who, and that gives you a snapshot of diet and time. We used stable isotopes, specifically nitrogen and carbon, to position and environmental signature of their prey. And for those of you who are isotope folks, we found specific isotope analysis so that we could actually share sites. So some of the questions we wanted to know were just, what's the variation in trophic niche space of giant trevally in the Western Indian Ocean? Are any differences driven by habitat? And then generally, how do the trophic positions of giant trevallies compare to other predators? So we collected samples from 4 sites over the course of 4 years, sampling 12 juveniles and 42 adults. 3 of those sites were in Seychelles, the first being Mahe, which was a granitic island site, and these samples were collected on coral and rocky reefs. And then Ryan Daly and with the help of Save Our Seas Foundation, collected samples which has these shallow sandbanks and flat reefs that encompass this lagoon, which supports coral reef and seagrass habitats. Then we have Providence Atoll. These samples were collected by Fly Castaway. And Providence is sandy, but lacking that elevated reef rock. It's also calcareous island and has these fringing barrier reefs. And then last but not least, we have a site in a kind of a continental coastal site in Mozambique, where again, Ryan Daly and others have collected sites. And this is interesting because this is actually a spawning aggregation. And so these fish are sort of a mix of resident and transient individuals. So jumping right into the results. So we have trophic position on the y-axis and carbon signature on the x-axis. And first of all, we've found an expanded trophic range of giant trevallies compared to previous studies. So we found some that have higher and lower trophic positions than other studies. And importantly, most of these are pretty high and actually at the equivalent level of many predatory tunas and sharks. Second, as you can see, giant trevallies exhibit complex trophic structure. So there's variation in their trophic position that's often, that in the Seychelles, actually throughout all these sites are really driven by carbon signature, ranging from this strong offshore signature in Ponta de Oro to a strong coastal signature in Providence, and not a whole lot of overlap between their trophic niches. So I mentioned we collected 12 juveniles. Those were all actually from, or for the most part, all from Mahe. And for juveniles, we found a strong correlation between fish size, fork length, which is shown on the x-axis, and trophic position, which is on the y-axis. So larger fish had higher trophic positions, but we did not see that in adult fish. And actually, what you can see is that across these sites, there's a lot of variation in trophic position amongst individual adults. And that is hinting at specialization in diet. And in fact, we think that's what's happening on St. Joseph Atoll. So for example, in St. Joseph, 2 of the largest individuals, which are shown here in black, have some of the lowest trophic positions. But these 2 individuals also happen to be equipped with long life acoustic transmitters. And this is work that Ryan Daly and others have just put out for a preprint. So check it out. And we know from these 2 fishes' movement behavior that they frequently visit this site, Marie Louis, which is about 90 kilometers away. And the other fish. And so this suggests that giant trevallies, or these two individuals anyway, have possibly developed local feeding strategies to utilize resources at neighboring islands. And I'll point out that this same kind of pattern and behavior has been observed in giant trevallies in Hawaii as well. So just a quick summary, this has been some really great work that I think has been especially cool because it's linked these different types of data, right? The genetics, the stable isotopes, and then a little bit of the movement behavior to really paint a clear picture of the evolution and ecology of these 2 species. And we're still continuing to work on both of these species, and I'm always open to collaborations on other trevally species in Seychelles and elsewhere. So just to give a quick recap, we found that both bluefin and giant trevallies were panmictic across the Seychelles and the Western Indian Ocean more broadly. However, I've highlighted these because to my knowledge, we're not really sure where they're actually spawning in Seychelles. And there's also been evidence of hybridization between both of these individuals. So if anyone knows information about this, I would love to hear from you because I'm also interested in doing some modeling and connectivity studies. And with the genomes, we're going to be looking at hybridization more closely too. As for diet of giant trevally, we found differences in trophic niches that are really driven by several factors, including changes, ontogenetic changes from juveniles to adults, which has to do with habitat usage, as well as some kind of hints at individual specialization in diet, but really a strong carbon signature that's driving differences in their trophic niches. And of course, they are top predators. And that was a really kind of, I think, impactful finding, because it shows that, in fact, these are really important components of the ecosystem in the Western Indian Ocean. Okay, so with that, there's a lot of funding sources and co-authors and project partners to thank, and including a huge list of the citizen scientists and sport fishermen and several other individuals that have been critical to completing this work. And like I said, it's still ongoing. And so with that, I thank you all for your attention. My video is stopped, but I'm going to try to start it again. If you have any questions, and if there's time, you can ask now or send me an email. Thank you.
All right, so thank you very much, Jessica. And the floor is now open for any questions. So I will just go through the questions. The first question is, do you think a loss of those species could lead to similar top-down trophic collapses as the loss of shark species?
Yeah, I think that these giant trevallies are less studied, certainly than shark species, and there's not information on, to my knowledge, in the Western Indian Ocean anyway, of their kind of prey consumption. But I know that there's been a lot of work in Hawaii on this, and the estimated numbers of like tonnage of year of prey species that both giant and bluefin trevallies eat is huge. And so certainly if these if their numbers go down, you could potentially see big shifts in prey species abundances as well. But yeah, that's a good question. I don't know if there's, yeah.
Thanks, Jessica. The next question is there any presence of the species in the Caribbean?
Nope, not these two species. There's other trevally species that are quite large that are found in the Caribbean, but these have a strict Indo-Pacific range. So the crevalle jack is a large trevally species that's looks similar to the GT, and it's found in the Caribbean and Atlantic.
So is there any research into the effect of catch and release on the behavior of these species?
Yes, I think there is some, I know there's been work done right on the skates and some other species. I'm not familiar with people that are doing catch and release mortality, but I know that there's been work done previously on this, like in Alphonse and possibly Farquhar. Someone asked if I saw there was a question that asked if there's opportunities to continue this work in Seychelles, and absolutely, especially to involve students. It'd be great to do this, to look at the bludgers, which are even more important trevally species in the inner islands, and definitely to involve students. It's, you know, there's a lot of training opportunities, and if you're interested in that and expanding any sort of genetic connectivity studies in Seychelles, definitely reach out to me.
Okay, thanks. The first person who asked the question, I'll just repeat it. It was the question about, is your method able to identify the food source from anthropogenic environment? There's an explanation below to the question. So basically, it says food source from anthropogenic environment means the fish possibly obtained the carbon and nitrogen originated from waste, water, and garbage.
Right, that's a good question. I think that you could do that if there were strong signatures from certain like water contaminants, like you could look at mercury, and people have looked at mercury in giant trevallies and several fish species. That would probably be the easiest. It has to be, you know, an isotope that would show sort of contamination or strong evidence of human kind of input.
All right, thank you, Jessica. So we have 2 more questions, and then I will move on to the next presentation. So the next one is, can you elaborate a bit more on the evidence for the hybridization between the species?
Yeah, so that's a great question, and I'm really excited about continuing that work in the future. So far, the estimate, and most of the information we have is from Hawaii, but the estimates are that they might hybridize at a rate of about 4%, which is pretty significant. And so the evidence we have so far is just from like a handful of both genetic data, which is pretty sparse. I actually have a bunch of samples that are likely hybrids as well that I haven't really had a chance to look at yet. But then there's been, you know, photos that are clear hybrids too, and there's been a lot of that's kind of what I'm hoping to get more of, is just from fishermen and other people that see a fish that doesn't really look quite like a GT or quite like a bluefin. And some of my samples, even from Seychelles, look to be hybrids. So I'm going to be exploring that more.
Thank you, Jessica. Okay, the next question is from Shane. So do you plan to conduct a similar research in other outer islands, such as Farquhar?
I'm not sure if that was in reference to the genetics or the isotopes, but I have genetic samples from Farquhar for both of the species, and we didn't, because they're all so connected, you know, for the purpose of this project, I was interested in like their broad patterns, but there could be potential genetic studies, particularly looking at like really fine scale connectivity between the islands. And similarly for the isotope and trophic position, it would be great to get more samples and to look at this more in depth, particularly the bluefin trevally, because there's some really great local knowledge from the fishing guides about bluefin trevally diet in Farquhar. It's just challenging to collect the isotope samples from several different locations because they have to be kept frozen, and there's a little bit more logistics. But yeah, it would be great to population connectivity. Okay, great, perfect. Glad I answered the question.
All right, Jessica, thank you very much for the great presentation, and to everybody who asked the questions.